Good morning. Welcome, Shama Stars. We are starting with new chapter 13, photosynthesis in higher plants. In this presentation, we will talk about early experiments done by various scientists to understand the process of photosynthesis. Photosynthesis is the process by which plants, some bacteria, and some protists convert light energy into chemical energy. They produce food for themselves and to feed the consumers who cannot make their own food. Oxygen, also essential for life, is a byproduct of this process, photosynthesis. To understand the process of photosynthesis, early experiments were done by Joseph Priestley, John Ingenhousz, Julius Vaughan, T.W. Engelmann, and Von Neal. We will discuss contribution of these scientists to understand the process of photosynthesis one by one. In 1970, Joseph Priestley performed a series of experiments that revealed the essential role of air in the growth of green plants. In his experiments, he observed that a candle burning in a closed space, a bell jar, soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed in air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Based on his observations, he concluded that plants restore to the air whatever breathing animals and burning candles remove. In 1796, Ingenhousz confirmed Priestley's finding. He discovered that plants release oxygen gas. He was the first to discover sunlight essential for photosynthesis and carbon dioxide used by plants. In 1854, a German botanist, Julius Vaughan, he is also known as the founder of experimental plant physiology, discovered that photosynthesis occurred in the chloroplast. He provided evidence for the production of glucose in the green parts of the plants and stored in the form of starch. Now consider the interesting experiments done by T.W. Engelmann. Using a prism, he split light into spectral components and then illuminated a green alga cladophora placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of oxygen evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. He first described the action spectrum of photosynthesis. The action spectrum resembles roughly the absorption spectrum of chlorophyll A and chlorophyll B. By the middle of the 19th century, the key features of plant photosynthesis were known, namely that plants could use light energy to make carbohydrates from carbon dioxide and water. A milestone contribution to the understanding of photosynthesis was made by a microbiologist, Van Neal, who, based on his studies of purple and green bacteria, demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. He gave the present day equation of photosynthesis. In green plants, water is the hydrogen donor and it is oxidized to oxygen. Some organisms do not release oxygen during photosynthesis. When hydrogen sulfide instead of hydrogen donor for purple and green sulfur bacteria, the oxidation product is sulfur or sulfate depending on the organisms and not oxygen. Hence, he concluded that oxygen evolved by the green plant comes from water, not from carbon dioxide. This was later proved by using radioisotopic techniques. So, this is all about the early experiments done by scientists to understand the process of photosynthesis. I hope you must have understood. In the next presentation, we will talk about where does photosynthesis take place and pigments involved in photosynthesis. Thank you, Shem Stars. That's all for today. You can drop your queries on Google Classroom. Take care.